In this video, I'm going to be showing you how DNA becomes inaccessible for gene expression. First, we look at our DNA molecule, which is these little strands of nucleotides wrapped around the proteins. And then those proteins are called histones, which they're just like the little 4 circle molecule thingies. And then DNA is wrapped around them. The histones help organize and package the DNA inside the cell. This structure can affect how accessible the DNA is to the gene reading machinery. And then another factor in making DNA inaccessible are methyl molecules. You can see there's one right there, and also right there. And these are small, like, chemical tags that attach to certain parts of the DNA. And when these methyl groups are added, they essentially lock the DNA, making it harder for the gene reading machinery to access it. This machinery, which includes proteins like RNA polymerase, is responsible for reading the DNA and creating messages that tell the cell how to build proteins. Right here we have our gene reading machinery. See some of the genes just go through that right there. And then the thing is, when these methyl molecules attach to the DNA, the machinery can't easily get in because the methyl is blocking it. And then this makes the gene inactive because it is not being read and translated into protein. So when DNA is covered with methyl molecules, the gene becomes inactive because the gene reading machinery can't access it. This is like a switch being turned off. On the other hand, when DNA is not methylated and it becomes more accessible, the gene is active and the machinery can read it, producing the necessary proteins. And then now let's answer a couple of important questions that were in the script I was told to answer. First one is, can the machinery read any significant stretch of DNA? And well, not always. The machinery can only read DNA when it's blocked by methyl groups or tightly wrapped around histones. Then number 2 is, would this be an active or inactive gene? And if the DNA is methylated, it would be an inactive gene because the machinery cannot access it. If there are no methyl groups blocking it, it would be active. So right here, this would be an inactive gene because there are methyl groups blocking the gene reading machinery from translating it into a protein.